Hello there, and welcome to the Cosmic Sleep Science. I am so glad you are here. Tonight, we drift into a world where color blooms beneath the surface of the sea. A place built not by hands, but by the quiet work of creatures smaller than your fingertip. Coral reefs hold secrets written in calcium and light, stories that unfold across centuries in waters that shimmer with life. Here, beneath the gentle rhythm of the waves, entire cities rise from the ocean floor. Towers of living mineral reach toward filtered sunlight. Shelters form in hidden chambers. And every surface tells a tale of patience, resilience, and the delicate dance between organisms that need one another to survive. If these peaceful explorations bring you comfort, I gently invite you to subscribe, share your thoughts, or simply rest here for a while. Your presence helps others discover this quiet corner of wonder. But for now, allow your shoulders to drop. Let the tension drift away from your body like sand settling through clear water. Breathe slowly and let us begin this tranquil journey together. Some coral polyps are so small that hundreds could rest on a single coin. These miniature architects possess bodies no larger than a pinhead, yet they accomplish extraordinary things. Each tiny creature extends delicate tentacles into the passing current, searching for microscopic meals that drift by in the darkness. Within their translucent tissues, they house even smaller partners, algae that capture sunlight and transform it into nourishment. Day after day, these minute beings deposit layers of mineral beneath themselves, building foundations that will outlast their own brief lives. The work happens slowly, almost invisibly, yet over years these pinpoint builders create structures vast enough to shelter thousands of species. Their size makes them vulnerable to countless threats, yet their numbers give them strength. A single coral head might contain tens of thousands of these gentle polyps, all connected, all working in quiet harmony beneath the waves. Some coral polyps are so small that hundreds could rest on a single coin. These miniature architects possess bodies no larger than a pinhead, yet they accomplish extraordinary things. Each tiny creature extends delicate tentacles into the passing current, searching for microscopic meals that drift by in the darkness. Within their translucent tissues, they house even smaller partners, algae that capture sunlight and transform it into nourishment. Day after day, these minute beings deposit layers of mineral beneath themselves, building foundations that will outlast their own brief lives. The work happens slowly, almost invisibly, yet over years these pinpoint builders create structures vast enough to shelter thousands of species. Their size makes them vulnerable to countless threats, yet their numbers give them strength. A single coral head might contain tens of thousands of these gentle polyps, all connected, all working in quiet harmony beneath the waves. Certain reefs shimmer, with colors that change as clouds pass overhead. The surface of a healthy coral colony reflects light in ways that shift from moment to moment. When the sun breaks through gaps in the clouds, greens deepen into emerald, purples brighten into violet, and yellows glow like captured sunshine. As shadows return, the colors soften into muted tones that blend with the surrounding water. This constant transformation comes from pigments within the coral tissues and the algae living inside them. Each species possesses its own palette, creating gardens of color that spread across the seafloor. The movement of water adds another layer of change, causing surfaces to ripple and flash as waves pass above. Standing before such a display feels like watching a living painting, one that breathes and shifts and never repeats itself exactly the same way twice. The beauty serves purposes beyond mere appearance, helping corals manage sunlight, attract certain organisms, and communicate their health to the ecosystem around them. Some reefs rest in waters so clear you can see 100 feet into the blue. In certain remote locations far from rivers and coastal disturbance, the ocean achieves a transparency that seems almost impossible. Sunlight penetrates deep into these crystalline waters, illuminating coral formations that extend down steep slopes and into shadowed valleys. Fish appear suspended in liquid glass, their every movement visible from remarkable distances. The clarity comes from the absence of sediments and nutrients that would cloud the water. Ironically, this purity creates challenges for life. since fewer nutrients mean less food drifting in the currents. Yet the corals here have adapted perfectly, relying heavily on their symbiotic algae and developing efficient ways to capture the sparse plankton that does appear. The visibility transforms, diving into an experience of floating through air rather than water. Formations that lie far below seem close enough to touch. The sense of space becomes vast and peaceful, a blue emptiness filled with scattered jewels of life clinging to stone. Certain coral species grow so slowly that a colony the size of a car might be older than a cathedral. In cooler waters or deeper zones where light barely reaches, corals add only a fraction of an inch to their structures each year. This patient growth means that some of the massive formations we encounter today began their lives centuries ago, long before modern cities existed. Their skeletons contain layer upon layer of mineral deposits, each one recording conditions that existed when it formed. These ancient colonies have witnessed countless storms, survived changing ocean temperatures, and persisted through periods when their surroundings transformed completely. Their longevity makes them living archives, storing information about past climates within their calcium carbonate bones. Scientists who study these slow-growing giants learn to read their layers like pages in a history book, discovering patterns that reveal how the ocean behaved long before instruments could measure it. The presence of such enduring organisms reminds us that some aspects of nature unfold across timescales far beyond human lifespans. Some coral polyps extend their feeding tentacles only when darkness falls. As daylight fades and the water dims to deep blue, certain corals undergo a quiet transformation. Structures that appeared smooth and compact during the day suddenly bloom into crowns of delicate arms. Each tentacle stretches outward, covered in specialized cells that detect and capture passing prey. The change happens gradually, a slow unfurling that responds to dropping light levels and shifting water chemistry. At night, the ocean fills with plankton that rise from deeper layers, creating a feast for organisms prepared to catch them. The coral polyps remain still, allowing currents to bring food to them, rather than expending energy in pursuit. When a tiny creature brushes against a tentacle, the response occurs in milliseconds. Stinging cells fire, threads deploy, and the prey becomes immobilized. The tentacle curls inward, guiding the captured meal toward a central mouth where digestion begins. By dawn, the tentacles retract, and the coral returns to its daytime appearance, having fed throughout the darkness. Certain reef formations create sounds that travel for miles underwater. The collective activity of countless organisms produces a symphony that fills the surrounding sea. Snapping shrimp create sharp cracks that echo off coral walls. Fish produce grunts, chirps, and grinding sounds as they feed and communicate. The movement of water through complex coral structures adds rushing whispers and gentle groans. Together, these sounds form a unique acoustic signature that identifies each reef as distinctly as a visual landscape. The noise carries far beyond the reef itself, traveling through water with surprising clarity. Young fish drifting in open ocean use these sound patterns to locate suitable habitats where they might settle and grow. The reef announces its presence to the surrounding sea, offering an auditory beacon that guides lost travelers home. Even in complete darkness, the sounds continue, creating an invisible map that animals follow with practiced ease. This underwater soundscape reveals a dimension of reef life that remains hidden from those who experience these ecosystems only through sight. Some corals release clouds of reproductive material that turn entire bays milky white. On specific nights each year, triggered by moon phases and water temperature, vast numbers of coral colonies spawn simultaneously. The coordinated release happens over just a few hours, transforming clear water into a blizzard of tiny bundles. Each package contains either eggs or sperm, floating upward toward the surface in slow motion. The water becomes so thick with genetic material that visibility drops to mere inches. Fish gather to feast on this abundance, creating a feeding frenzy that lasts through the night. Yet enough packages survive to ensure that fertilization occurs, with eggs and sperm from different colonies meeting in the open water. The timing must be perfect, since these reproductive cells remain viable for only a short period. The synchronization across an entire reef demonstrates a level of coordination that still puzzles researchers. Chemical signals, temperature cues, and light patterns all play roles in this underwater ballet. By morning, the clouds have dispersed, carried by currents across vast distances, where some will eventually settle and begin new colonies. Certain reefs shelter fish that change gender as they grow. In the complex social structures of reef communities, some species possess the remarkable ability to switch from female to male or male to female, depending on circumstances. A group of fish living among branching corals might consist entirely of females and one dominant male. When that male disappears, the largest female begins a transformation that restructures her entire body. Over weeks, her reproductive organs change, her coloration shifts, and her behavior adapts to her new role. The transition allows the group to maintain its social structure and continue reproducing despite the loss of the original male. This flexibility provides an evolutionary advantage in invanage, where finding new mates might be difficult. Some species can switch multiple times throughout their lives, responding to changing group dynamics. The process involves complex hormonal changes guided by social cues and environmental factors. These gender-fluid fish demonstrate nature's creative solutions to the challenges of survival and reproduction in the structured world of the reef. Some coral skeletons contain pores so tiny that thousands fit in a space smaller than a rice grain. The mineral structures built by corals are not solid masses, but intricate lattices filled with microscopic spaces. These pores create a skeleton that is both strong and light, able to withstand pressure while remaining less dense than solid stone. The network of spaces also provides homes for countless tiny organisms that burrow into the coral structure. Bacteria, algae, small worms, and other creatures colonize these chambers, forming hidden communities within the skeleton itself. The pores allow water to circulate through the coral's interior, bringing oxygen and nutrients to residents living deep within. As the coral grows, it continually creates new pores while sealing off others, maintaining a dynamic architecture that evolves over time. The complexity of this structure becomes apparent only under magnification, where what seemed like simple stone reveals itself as an elaborate maze of passages, chambers, and connecting tunnels. This hidden architecture contributes to the reef's overall stability and provides habitat far beyond the visible surface. Certain coral communities form partnerships with fish that never stray far from their branches. Small damselfish claim individual coral heads as territories. They defend vigorously against all intruders. These fish spend their entire lives within a few feet of their chosen coral, chasing away competitors and predators alike. The coral benefits from this protection, as the resident fish removes parasites and prevents other creatures from settling on its surface. The fish gains a safe home and often cultivates gardens of algae on nearby rocks, creating a personal food supply. The relationship becomes so specific that some fish species associate only with certain coral types, forming partnerships that persist across generations. Young fish inherit territories from their parents or compete for unclaimed corals, where they will spend their adult lives. The constant presence of these guardians influences the health and growth patterns of their coral hosts. Areas with active fish residents often show faster growth and fewer signs of disease. These miniature territories become the foundation of complex social networks that extend across the reef. Some reefs grow on slopes so steep that they resemble underwater cliffs. In certain locations, coral formations cling to near vertical walls that plunge from shallow waters into abyssal depths. These wall reefs create dramatic transitions between light and darkness, warmth and cold. The upper sections bask in bright sunlight, hosting corals that spread into plates and tables to capture maximum illumination. As the wall descends, light fades and different species appear, adapted to dimmer conditions. Eventually, the slope enters perpetual twilight where only the hardiest corals persist, their growth slowed to a crawl by the scarcity of light. Divers exploring these walls experience the reef as a vertical journey through changing communities. Fish school along the face of the cliff, using it as a highway between deep feeding grounds and shallow shelters. Currents strike these walls with force, bringing nutrients from the depths and sweeping them past waiting polyps. The sensation of hovering beside such a formation, with bright water above and darkness below, creates a profound sense of the ocean's vertical dimension. Certain corals build structures that channel water into predictable patterns. The shape of coral colonies influences how currents move across the reef. Branching formations create sheltered pockets where water slows and eddies form. Massive round corals deflect flow around their sides, creating zones of calm in their wake. Plate corals act like roofs, directing water beneath them through narrow gaps. These patterns are not random but result from the coral's growth responding to water movement. Over many years, the structures evolve to optimize food delivery, bringing plankton, rich currents past feeding polyps, while preventing sediment from settling on delicate tissues. Other reef inhabitants learn to read these flow patterns, positioning themselves in areas where food concentrates, or where gentle currents provide relief from strong surge. The interaction between living structure and moving water creates a dynamic environment that shifts with tides and seasons. During storms, these same patterns help dissipate wave energy, protecting the reef from catastrophic damage. Some coral polyps communicate with neighbors through electrical signals. Beneath the visible surface, tiny currents flow between connected polyps, carrying information about threats, food availability, and environmental changes. When one polyp encounters something harmful, it sends signals that cause neighbors to retract their tentacles or increase mucus production. This coordinated response happens faster than chemical communication alone could achieve. The electrical impulses travel through tissues that connect all polyps in a colony, essentially creating a living network. During feeding, signals help synchronize tentacle movements, improving the colony's ability to capture prey. The system allows corals to behave as unified organisms, despite being composed of thousands of individuals. Recent research has revealed that these signals vary in strength and pattern, depending on the stimulus, suggesting a more complex communication system than previously understood. The discovery adds another layer to our appreciation of coral intelligence, showing that even organisms without brains or nervous systems can process information and coordinate responses with remarkable sophistication. Certain reefs contain caves, where water changes temperature by seasons alone. In sheltered grottos and deep overhangs, water becomes isolated from the surrounding ocean for extended periods. During summer, these pockets warm gradually, creating tropical microclimates within otherwise temperate reefs. In winter, they cool slowly, maintaining stable conditions, even as outside temperatures fluctuate. Corals growing in these caves experience very different environments than those on exposed surfaces just a few feet away. Some species use these refuges to survive beyond their normal geographic ranges, persisting in waters that would otherwise be too cold or too variable. The caves also shelter organisms during their most vulnerable life stages, providing nursery areas where young corals and fish develop protected from predators and harsh conditions. The existence of these thermal refuges becomes increasingly important as ocean temperatures shift globally. They offer glimpses of how reefs might adapt to changing climates by finding and utilizing stable pockets within otherwise challenging environments. Some coral larvae possess sensory abilities that guide them to perfect settlement spots. These tiny drifting forms, barely visible to the naked eye, can detect chemical signals released by healthy reefs. They sense the texture of surfaces through specialized cells, distinguishing between suitable substrates and unsuitable ones. Light levels inform them about depth, helping them avoid areas too deep for their algae partners to function effectively. Even sound plays a role, as larvae move toward the crackling symphony of healthy reefs and away from the silence of degraded areas. This sensory sophistication allows them to make critical decisions about where to begin their adult lives. Once a suitable spot is found, the larva attaches permanently, cementing itself to the surface and beginning its transformation into a polyp. The location chosen in these first moments determines the coral's entire future, influencing how much light it will receive, how strong the currents it must withstand, and which neighbors it will grow beside. The ability of such small, simple organisms to assess and select their homes reveals unexpected complexity in the earliest stages of coral life. certain coral reefs support more biomass than equivalent areas of farmland. The density of life within a thriving reef ecosystem can exceed what terrestrial environments produce in similar spaces. Fish, invertebrates, algae, and the corals themselves create a living mass that fills every available niche. The three-dimensional structure of the reef allows organisms to stack vertically, with different species occupying layers from the seafloor to several meters above it. This architectural complexity enables more life to coexist than would be possible on a flat surface. Energy flows through the system with remarkable efficiency, as waste from one organism becomes food for another. The warm water and abundant sunlight of tropical reefs accelerate metabolic processes, allowing rapid growth and reproduction. Predators, grazers, filter feeders, and decomposers all find roles within the community, creating a balanced system that supports exceptional productivity. The comparison to farmland becomes even more striking when considering that reefs achieve this abundance in nutrient-poor waters, relying on efficient recycling rather than external inputs. Some corals extend delicate filaments to digest competitors that grow too close. When two coral colonies expand until they touch, territorial battles sometimes occur at the microscopic level. Certain species deploy special digestive filaments from their mouths that extend outward and digest the tissues of neighboring corals. These filaments release enzymes that break down the competitor's flesh, creating space for the attacking colony to expand. The battles unfold slowly, over days or weeks, as each colony attempts to dominate the contested border. Other species rely on faster-growing edges that simply overgrow their neighbors, shading them from sunlight. Some produce chemical compounds that inhibit nearby competitors without physical contact. These interactions create invisible boundaries across the reef, determining which species succeed in different areas. The outcomes depend on species identities, environmental conditions, and sometimes random factors like which colony was established first. Over time, these small territorial disputes shape the composition and appearance of entire reef sections. Certain fish remove parasites from corals during daily cleaning sessions. Small wrasses and gobies patrol coral surfaces, carefully picking at the tissue to remove unwanted organisms. The corals seem to tolerate this activity, perhaps even benefiting from the removal of irritating parasites. The fish gain nutrition from the creatures they consume, while the coral enjoys relief from organisms that might otherwise cause damage. These cleaning behaviors occur at predictable times and locations, with some fish maintaining regular routes across specific coral colonies. The relationship demonstrates cooperation between very different organisms, each receiving benefits from the interaction. Some fish species specialize in cleaning particular coral types, developing techniques suited to specific growth forms or tissue textures. The cleaning sessions can last several minutes, with the fish working methodically across the coral surface. Other reef inhabitants learn to recognize these cleaning sites and gather nearby, creating small communities focused around the mutual benefits of parasite removal. Some coral colonies grow in perfect circles that expand outward from a central point. These ring-shaped formations begin when a small fragment settles and starts growing. As the colony expands, the center ages and eventually dies, while the outer edge continues to grow vigorously. Over decades, this pattern creates a ring that slowly increases in diameter. The dead center sometimes fills with sediment or becomes colonized by other organisms, creating a distinct zone within the circle. The rings can grow large enough to be visible from above the water, forming features in the landscape of shallow reefs. Their circular shape influences water flow in ways that benefit the living rim, concentrating food delivery along the active edges. Other organisms recognize these formations as stable structures and settle preferentially along the rings, creating communities shaped by the coral's growth pattern. The perfect geometry of these natural circles demonstrates how simple growth rules, applied consistently over long periods, can create striking patterns. Certain reefs contain coral gardens, where every surface blooms with color. In locations with perfect combinations of light, current, and temperature, corals achieve a density and vibrancy that exceeds what seems possible. Every available space becomes occupied by living tissue in shades of purple, orange, pink, green, and blue. The gardens appear almost artificial in their abundance, challenging expectations of what natural growth should look like. Multiple species grow intermingled, creating tapestries where branching corals rise from fields of encrusting forms. The color comes from both the corals themselves and the algae within them, with each species contributing its own hues to the overall palette. Fish moving through these gardens appear drab by comparison, their colors muted against the vibrant backdrop. The existence of such gardens depends on stable conditions remaining constant for many years, allowing corals to reach maximum health and coverage. Even small disturbances can reduce the vibrancy of these special places, making them indicators of overall reef health. Some coral polyps can retract completely into their skeletons in seconds. When danger approaches or conditions suddenly deteriorate, certain corals pull their soft tissues deep into protective cups within their mineral structures. The transformation happens almost instantly, triggered by touch, shadows, or chemical signals. One moment, the coral appears fully extended and active. The next, it becomes a lifeless-looking stone surface. The retraction protects delicate tissues from predators, strong surge, or excessive sunlight. Once the threat passes, the polyps slowly re-emerge, extending tentacles and resuming normal activities. This defensive ability varies among species, with some corals remaining highly sensitive to disturbance, while others ignore all but the most severe threats. The speed of the retraction relies on muscles within each polyp that contract rapidly, pulling tissue inward while simultaneously forcing water out of internal spaces. The polyps can remain retracted for extended periods if necessary, though doing so prevents feeding and requires energy to maintain. Certain corals build branches that spiral like underwater cinnamon sticks. The growth patterns of some species create twisted formations that coil as they extend upward. This spiraling results from uneven growth rates on different sides of the branch, with one edge growing slightly faster than the other. Over time, this subtle difference creates a corkscrew pattern that winds through the water. The spirals may help strengthen the branch against breaking, distributing stress along the twisted structure. They also create interesting flow patterns as water moves through and around the coils. Small organisms find shelter in the gaps between spirals, using the three-dimensional structure as protection. The formations vary in tightness, with some displaying gentle twists and others coiling in tight helixes. The visual effect creates an almost artistic quality, as if the corals were deliberately sculpted rather than grown through biological processes. These spiraling branches add complexity to the reef architecture and provide unique microhabitats unavailable in straight-branched species. Some reefs pulse with bioluminescent waves when disturbed at night. In darkness, certain organisms release glowing clouds when touched or startled. A hand or fin passing through the water triggers cascades of blue-green light that ripple outward in expanding circles. The effect comes from dinoflagellates, single-celled organisms that produce light through chemical reactions. During the day, they remain dormant, but at night they transform the water into a liquid that glows at the slightest disturbance. Fish swimming through these areas leave trails of light in their wake, their movements traced in luminous lines. Waves breaking over the reef crest create glowing foam that sparkles and fades. The phenomenon serves as a defense mechanism for the dinoflagellates, as the sudden light startles predators or attracts larger hunters to the area. For humans witnessing this display, the experience feels magical, turning every movement into an artistic gesture painted in living light. Certain coral species host communities of worms that never see daylight. Within the labyrinth of coral branches and crevices, countless worms make their homes in permanent darkness. These creatures possess bodies adapted to life in tight spaces, with elongated forms and reduced eyes. Some feed on particles that settle into the coral structure, while others filter tiny organisms from water flowing through hidden channels. The worms contribute to the reef ecosystem by breaking down organic material and recycling nutrients within the coral framework. Many species spend their entire lives within a single coral colony, reproducing and raising young in the same dark chambers their parents occupied. The diversity of these hidden worms often surprises researchers who carefully examine coral structures. Dozens of species might inhabit a single large colony, each occupying its own preferred depth and location within the maze. Some corals adjust their skeleton density based on wave strength. In areas exposed to strong surge, certain species build thicker, denser skeletons that resist breaking. The same species growing in calm lagoons produces lighter, more porous structures since heavy reinforcement becomes unnecessary. This plasticity allows corals to optimize their construction for local conditions, investing resources in strength only where needed. The adjustment happens gradually as the coral grows, with each new layer reflecting current wave conditions. Colonies relocated from rough water to calm areas begin producing lighter skeletons over time, demonstrating the responsiveness of this adaptation. The ability to fine-tune structural strength shows how corals integrate environmental information into their growth patterns, creating architectures perfectly suited to their specific locations. Certain reef fishes remember the locations of hundreds of individual corals. In their daily movements, these fish navigate complex three-dimensional landscapes using spatial memories of remarkable detail. They recall which coral heads provide the best shelter during storms, where cleaning stations operate, and which structures host their preferred prey. This cognitive mapping allows efficient movement through the reef, conserving energy and reducing exposure to predators. Young fish learn these routes through exploration and by following experienced individuals. Over time, they build mental maps that cover their entire home range. Changes to familiar coral structures, such as storm damage or new growth, get incorporated into these maps through continued observation. The sophistication of these navigational abilities suggests that reef fish possess cognitive capacities more advanced than traditionally assumed for such small-brained creatures. Some coral polyps possess tentacles covered in thousands of microscopic weapons. Each tentacle bristles with specialized cells called cnidocytes, containing miniature harpoons coiled under pressure. When triggered by contact or chemical signals, these cells explode, launching their barbed threads in less than a millisecond. The threads pierce prey and inject toxins that immobilize or kill. Each polyp might possess hundreds of tentacles, and each tentacle contains thousands of these cellular weapons, creating an arsenal of devastating efficiency. After firing, the spent cells are absorbed and replaced with new ones, maintaining the coral's defensive and predatory capabilities. Different coral species produce different toxins, some targeting specific prey types. The weapons work on extremely small scales, effective against creatures measured in millimeters. Yet the cumulative effect allows corals to defend themselves and feed successfully in the competitive reef environment. Certain corals create mucus that acts as sunscreen for their tissues. In shallow waters where ultraviolet radiation penetrates strongly, some species produce protective slime that absorbs harmful wavelengths before they reach sensitive cells. The mucus contains compounds similar to those used in human sunscreens, filtering out damaging light while allowing useful wavelengths to pass through to algae partners. The thickness of this layer varies with light intensity, increasing during periods of strong sunshine and thinning when clouds provide relief. This chemical sunscreen works alongside other protective mechanisms, including pigments within the coral tissue itself. Together, these defenses allow corals to thrive in environments where UV exposure would otherwise cause severe damage. The discovery of these natural compounds has inspired research into developing more effective and environmentally friendly sunscreen products for human use. Some reefs develop pools that trap water during low tides. On flat reef platforms exposed to air at the lowest tides, water remains in depressions, creating temporary isolated habitats. These tide pools contain miniature reef communities that endure rapidly changing conditions. As the sun beats down on the trapped water, temperatures rise dramatically. Evaporation increases salinity beyond ocean levels. Oxygen concentrations fluctuate wildly as organisms consume it faster than waves can replenish it. Yet corals and other creatures surviving in these pools have adapted to extremes that would kill their relatives just a few meters away in permanently submerged areas. When the tide returns, the pools reconnect with the sea, flushing out accumulated heat and chemicals. The organisms endure this cycle daily, demonstrating remarkable physiological flexibility. These pools serve as natural laboratories for studying how reef creatures might cope with future ocean changes. Certain coral larvae can delay settlement for weeks until perfect conditions appear. Instead of attaching to the first suitable surface they encounter, some species remain in their larval form, drifting and searching for optimal locations. This extended competency period requires energy reserves and exposes the larvae to predators, but increases chances of finding ideal settlement sites. The larvae use their sensory abilities to continually assess their surroundings, rejecting locations that fail to meet their requirements. Only when all factors align, correct substrate, appropriate depth, healthy reef sounds, and smells does settlement occur. This selectivity helps explain why some reefs recruit new corals successfully, while nearby areas remain barren despite receiving similar numbers of larvae. The ability to delay settlement represents a bet that the risks of extended drifting are worth the benefits of choosing wisely. Some corals survive burial under sand by extending tissues upward through the sediment. When storm waves deposit layers of sand over coral colonies, certain species respond by growing their tissues vertically, maintaining contact with the water above. Polyps at the top of the buried colony stretch upward, while those completely covered enter a dormant state. The colony sacrifices energy to maintain this connection, but avoids suffocation and can resume normal function once currents remove the sand. Not all species possess this ability. Those that cannot escape burial often die. The capacity to survive sand inundation determines which corals succeed in environments where sediments shift frequently. Over time, reefs in sandy areas become dominated by species with these escape abilities, creating communities adapted to unstable substrates. Certain fish use coral branches as percussion instruments during courtship. Males of some species grasp coral structures with their mouths and vibrate rapidly, creating clicking sounds that travel through the reef. These acoustic signals attract females and warn rival males away from territories. The hardness and shape of different corals produce distinct sounds, allowing fish to choose instruments that create their preferred tones. Some males develop sight fidelity to particular coral heads, whose acoustic properties suit their displays. The repeated drumming does not appear to harm the corals, though polyps may retract temporarily in response to the vibrations. This behavior demonstrates another way reef inhabitants use coral structures as tools, integrating the physical environment into their reproductive strategies. Some coral colonies show preferences for certain orientations toward current and light. Given choices during early growth, individuals of many species position themselves to maximize food delivery, while optimizing light capture for their algae partners. Branches extend preferentially into prevailing currents that bring plankton. Plates angle to catch morning or afternoon sunlight depending on local conditions. These orientations are not random, but reflect sensory feedback that guides growth patterns. Colonies that achieve optimal orientations grow faster and reproduce more successfully than poorly positioned individuals. The ability to sense and respond to directional cues shows that corals actively engage with their environment, rather than growing passively according to fixed genetic programs. Certain reefs contain coral formations that resemble human-made sculptures. Some massive corals develop shapes that look deliberately carved, with smooth curves, balanced proportions, and striking symmetries. These forms arise from the coral's growth patterns, interacting with local water movement, creating natural art through biological and physical processes. Other species produce irregular, chaotic structures that resemble abstract sculptures. The variety of forms across a reef creates visual diversity that changes the character of different areas. Some formations become iconic landmarks within their reefs, recognized by divers and fish alike. The fact that living organisms can create structures of such aesthetic appeal through simple growth rules highlights the unexpected beauty that emerges from natural processes. Some coral polyps can sense the approach of predators before physical contact occurs. Certain species detect chemical cues released by approaching starfish or snails that feed on corals. When these signals reach the polyps, they trigger defensive responses, including increased mucus production and tissue retraction. Some corals even alter their growth patterns to make themselves less attractive to specific predators. This chemical awareness provides an early warning system that improves survival. The sensitivity varies among species, with some responding to incredibly dilute concentrations of predator chemicals, while others react only to strong signals. This variation reflects different evolutionary histories and predator pressures across reef environments. Certain coral branches grow at angles that minimize damage from falling debris. In reef areas where fragments regularly break off overhead formations, some species develop growth patterns that deflect falling material rather than catching it. Branches angle to guide debris away from the colony center, protecting the most vital tissues. This adaptive architecture reduces casualties from routine disturbances that affect all reefs. The angles adjust over time as the coral responds to actual damage patterns, essentially learning from experience through modification of growth directions. This responsive architecture shows how coral forms reflect not just generic species traits, but specific local conditions. Some reefs contain corridors that different fish species use at different times. Certain pathways through the coral structure serve as highways during dawn migrations, become hunting grounds at midday, and transform into shelter zones at dusk. The same physical space serves multiple functions as different species cycle through their daily routines. This temporal sharing allows more species to coexist than the reef could support if each required exclusive territory. The timing of these transitions is remarkably consistent, suggesting that fish possess accurate internal clocks synchronized to light cycles and tidal patterns. Certain corals produce chemicals that attract specific species of protective shrimp. These compounds act as chemical advertisements, signaling to shrimp that the coral offers suitable habitat. The shrimp respond by settling preferentially on corals, producing the strongest signals. Once established, the shrimp defend the coral from predators, creating a mutualistic relationship that benefits both partners. The specificity of these chemical signals helps explain why certain coral-shrimp partnerships are so consistent across geographic regions. Both partners have evolved to recognize each other through these molecular handshakes. Some coral colonies develop asymmetrical forms in response to consistent wave direction. One side of the colony might extend far from the base, while the opposite side remains compact. This lopsided growth positions feeding polyps optimally in the current, while minimizing the colony's profile to reduce wave stress. The asymmetry creates distinctive shapes that reveal prevailing water movement patterns. Experienced observers can reconstruct local current regimes by examining coral growth forms, reading the physical environment through the living architecture of the reef. Certain reef environments contain more diversity in a cubic meter than some entire ecosystems. The concentration of species within small volumes of reef habitat can exceed what forests, grasslands, or other terrestrial ecosystems achieve. Every surface, crevice, and water layer contains organisms specialized for that precise microhabitat. This extreme diversity arises from the three-dimensional structure corals create, the stability of tropical conditions, and millions of years of evolution in these environments. The result is a biological treasure that continues to reveal new species and relationships as researchers develop better tools for observation. Some corals extend their polyps in rhythmic patterns that create waves across the colony surface. These coordinated movements may enhance water flow over the colony, improving nutrient delivery and waste removal. The waves of extension and retraction create visual patterns that ripple across the coral surface, visible to observers who watch patiently. The purpose of this coordination is not fully understood, but it demonstrates communication and collective behavior within the colony. Individual polyps somehow synchronize their actions, creating emergent patterns from thousands of individual movements. Certain fish remove algae from coral surfaces with surgical precision. These careful grazers distinguish between harmful algae that compete with corals and beneficial films that provide services. They crop unwanted growth without damaging coral tissue, maintaining the delicate balance that keeps corals healthy. The precision of this grazing requires sensory abilities that distinguish between different algae types by texture, chemistry, or appearance. The fish essentially garden the corals, curating the community of organisms on their surfaces in ways that benefit both grazer and coral. Some coral reefs create eddies that concentrate plankton in specific zones. The large-scale structure of entire reef systems influences water movement in ways that trap food in predictable locations. Fish learn these zones and gather where the reef itself concentrates their meals. The effect amplifies during certain tidal phases when currents interact most strongly with reef topography. Understanding these patterns helps explain why particular reef areas support higher fish densities than others with apparently similar coral cover. Certain corals can partially digest the algae living in their tissues. When nutrients become extremely scarce, some species begin breaking down portions of their algae population, consuming them for energy. This cannibalistic behavior provides emergency nutrition during stressful periods, but weakens the partnership that normally sustains the coral. The balance between maintaining adequate algae and harvesting them for food represents a delicate calculation that affects long-term survival. Corals that digest too many algae lose their primary energy source, while those that maintain full populations may starve during extended nutrient scarcity. Some reef channels accelerate water to speeds that prevent small organisms from maintaining position. These natural flumes create barriers that separate reef zones as effectively as land barriers divide terrestrial habitats. Species unable to cross the fast-flowing channels remain isolated in their respective zones, even though they could easily swim the distances involved under calm conditions. The channels influence community composition by determining which species can move between areas and which remain confined to specific sections. Certain coral polyps retract at different speeds, depending on threat level. Gentle touches trigger slow withdrawals that conserve energy, while sudden shadows or strong disturbances cause rapid retraction. This tiered response system allows corals to react appropriately to various dangers without wasting resources on unnecessary emergency responses. The ability to assess threat levels suggests sensory processing more sophisticated than simple reflex reactions. Polyps integrate information from multiple stimuli before determining appropriate response intensity. Some corals grow faster on their shaded sides than in direct sunlight. This counterintuitive pattern occurs when light becomes excessive, stressing tissues and slowing growth. By extending into shaded zones, the coral finds more favorable conditions for the partnership with its algae. The resulting growth form may appear lopsided, but it represents an optimal response to local light conditions. This behavior challenges assumptions that corals always seek maximum sunlight exposure. Certain reef fish have learned to farm algae on dead coral skeletons. These agricultural fish maintain gardens on specific pieces of rubble, defending them aggressively and managing the algae through selective grazing. The cultivated plots provide more nutrition than random grazing would yield, giving farming fish advantages during seasons when food becomes scarce. The behavior requires memory, territorial defense, and understanding of cause and effect, cognitive abilities once thought limited to more complex animals. Some coral colonies develop color patterns that differ between their sunlit and shaded sides. The variation reflects different pigment concentrations needed to manage light exposure optimally. Sunlit areas might appear pale or fluorescent to reflect excess light, while shaded zones develop darker colors to capture available photons more efficiently. These within-colony variations show that corals don't simply express fixed color patterns, but actively adjust pigmentation to local conditions across their surfaces. Certain corals produce antibacterial compounds that keep their surfaces remarkably clean. These chemical defenses prevent harmful bacteria from colonizing coral tissues and potentially causing disease. The compounds vary among species, with each producing its own suite of protective molecules. Some of these natural antibiotics have attracted interest from pharmaceutical researchers seeking new medicines. The corals continuously produce and release these compounds, maintaining a chemical shield that operates even while the coral sleeps. Some coral polyps coordinate their feeding to create currents that benefit the entire colony. When thousands of polyps beat their cilia in synchronized patterns, they generate water flow that extends beyond individual reach. This collective pumping brings fresh nutrients across the colony surface and carries waste away more efficiently than passive currents alone could achieve. The coordination requires no central control, yet emerges from simple rules followed by each polyp. The result is a self-organizing system that improves feeding efficiency for all members. During calm periods when ocean currents slow, this internal pumping becomes especially important, allowing the colony to maintain function even in still water. Certain reef crabs decorate themselves with living coral fragments for camouflage. These industrious crustaceans carefully select small pieces of coral and attach them to their shells using specialized hooks on their backs. As the coral fragments continue growing, they create living disguises that make the crabs nearly invisible against reef backgrounds. The crabs maintain their decorations, replacing dead fragments with fresh ones and adjusting their camouflage as they move between different reef zones. This behavior requires the crab to distinguish between coral types and understand which fragments best match their current surroundings. The partnership sometimes benefits the coral too, as fragments that might otherwise die gain mobility and access to new locations. Some corals release defensive chemicals when their neighbors die, detecting the chemical signals of coral death. Nearby colonies increase production of protective compounds and strengthen their immune responses. This preemptive defense helps them resist whatever killed their neighbors, whether disease, predation, or environmental stress. The ability to sense and respond to distant threats shows that coral colonies communicate across gaps of open water. The chemical warnings spread through the reef like invisible alarm signals, preparing entire communities for incoming danger. Certain fish perform dances that signal their intentions to coral-dwelling partners. Before entering the territory of aggressive damselfish that guard coral heads, some species perform specific swimming patterns that identify them as cleaners or harmless visitors, rather than threats. The damselfish recognize these signals and allow passage to dancers while attacking others who approach without proper introduction. These ritualized behaviors reduce conflict and allow beneficial interactions to occur. The dances vary between regions, suggesting learned behaviors passed between generations, rather than purely instinctive responses. Some coral colonies develop multiple color morphs within a single genetic individual. Different branches of the same colony may display purple, green, and brown colors simultaneously, each responding to the specific light and flow conditions it experiences. This within-colony variation shows the remarkable plasticity of coral physiology. The same genetic code produces different appearances depending on environmental inputs, creating colonies that appear to be patchworks of multiple species. The variation serves functional purposes, optimizing each section for its particular microenvironment. Certain reef sponges create currents stronger than the coral polyps around them. Despite having no muscles or moving parts visible to the eye, sponges generate powerful water flow through their bodies. Microscopic flagella beat in coordination within the sponge's internal chambers, creating suction that pulls water through elaborate filter systems. A single large sponge might pump thousands of liters daily, filtering particles and processing nutrients at rates that influence the entire surrounding area. The current they create benefits nearby corals by bringing fresh water and carrying waste away from reef surfaces. Some corals survive complete burial by entering dormancy states lasting months. When sediments cover them entirely, these species shut down most metabolic processes, entering suspended animation until conditions improve. They survive on stored energy reserves, waiting in darkness until currents shift the covering sediments. Once exposed again, they resume normal function, often with minimal tissue loss. This remarkable ability allows them to inhabit environments too unstable for less adaptable species. The dormancy mechanism involves complex cellular changes that protect tissues from damage during the long periods without light or normal water flow. Certain fish eggs attach to coral branches where parents guard them until hatching. The coral provides elevation above the seafloor where sediments might smother eggs. and its complex structure offers defensive positions for parent fish. The eggs develop in relative safety, protected by both coral architecture and parental vigilance. After hatching, larvae often remain near the natal coral for days, using its shelter before dispersing into open water. This reproductive strategy creates strong associations between specific fish species and particular coral types, with some fish refusing to spawn anywhere except on their preferred coral species. Some coral mucus contains sugars that feed beneficial bacteria living on reef surfaces. These bacteria form protective biofilms that help prevent pathogenic organisms from colonizing coral tissues. The coral essentially cultivates bacterial gardens by providing specific nutrients that favor helpful species over harmful ones. This manipulation of microbial communities represents a sophisticated immune strategy that operates continuously. The balance of bacterial species shifts in response to environmental stress, with the coral adjusting its mucus composition to encourage bacteria that provide the greatest benefits under current conditions. Certain corals build skeletons with seasonal bands visible under X-ray imaging. Like tree rings, these bands record growth patterns and environmental conditions year by year. Dense bands form during favorable seasons, while lighter bands mark periods of stress or slow growth. Researchers extract cores from long-lived colonies and analyze these bands to reconstruct ocean conditions, extending back centuries. The records reveal patterns of temperature change, storm frequency, and oceanographic shifts that occurred long before modern monitoring began. Each coral becomes a climate archive, storing data within its bones that tells stories of past seas. Some reef sharks follow the same patrol routes so consistently that other fish anticipate their appearances. Prey species clear from the sharks' paths moments before they arrive, responding to time cues rather than direct sensory detection. The regularity of these patrols suggests that sharks use temporal as well as spatial memory, following schedules that optimize hunting efficiency. Other reef inhabitants synchronize their activities to these predictable patterns, feeding in safe periods and sheltering when sharks patrol nearby areas. Certain corals extend special sweeper tentacles, much longer than normal polyp structures. These aggressive tentacles reach toward neighboring corals, stinging and damaging competitors that grow too close. The sweeper tentacles may extend several inches from the colony, creating a defended zone that prevents overgrowth by faster-spreading species. The tentacles emerge only when the coral detects nearby competition, showing that their development is triggered by environmental cues rather than constant growth patterns. This defensive strategy requires energy investment, but preserves space critical for long-term survival. Some coral larvae preferentially settle near adults of their own species. Chemical cues released by established colonies help larvae identify suitable habitat where their species already succeeds. This preference leads to clustering patterns where certain reef areas become dominated by single species. The strategy makes sense because adult presence indicates favorable conditions, though it also creates vulnerability since disturbances can eliminate entire local populations. The larvae must balance the benefits of settling near kin against the risks of concentrating in limited areas. Certain fish have evolved immunity to specific coral stings. These species move freely among tentacles that would paralyze other fish, using the coral's defenses as protection from their own predators. The immunity develops through special mucus coatings or cellular adaptations that neutralize the coral's toxins. Some fish are born with these protections, while others acquire them gradually through careful exposure. The relationship benefits fish dramatically, but provides less obvious advantages to corals, creating an asymmetrical partnership. Some corals reproduce by fragmenting during storms, turning destruction into opportunity. Branches broken by waves don't die but become new colonies if they land in favorable locations. This asexual reproduction supplements sexual spawning, allowing rapid expansion after disturbances. Species employing this strategy often dominate areas with frequent storms, their fragmentation biology turning environmental challenges into competitive advantages. The fragments require no time for larval development or settlement, beginning growth immediately once they stabilize on the substrate. Certain coral colonies show wound healing rates that vary seasonally. During warm months when energy is abundant, injuries close quickly with new tissue covering exposed skeleton within days. In cooler seasons or during stress periods, healing slows dramatically, leaving wounds exposed for weeks. The seasonal variation in healing capacity affects vulnerability to disease, as open wounds provide entry points for pathogens. Corals damaged during unfavorable seasons suffer higher mortality than those injured when conditions support rapid tissue regeneration. Some reef octopuses learn to open specific coral crevices to access hidden prey. These intelligent hunters remember which coral formations contain shrimp or crabs and develop techniques to extract them. The octopuses manipulate coral branches with their arms, prying apart structures or reaching into narrow gaps. They return repeatedly to productive hunting sites, suggesting memory of both locations and techniques that worked previously. This problem-solving ability demonstrates cognitive sophistication unexpected in invertebrates. Certain corals maintain backup populations of symbiotic algae in their deep tissues. When surface algae die from stress, these deep reserves survive and can repopulate the colony once conditions improve. This biological insurance policy helps corals survive bleaching events that kill their primary algae populations. The backup algae often belong to more heat-tolerant types, providing the coral with different options for rebuilding symbioses. The strategy shows that corals actively manage multiple algae strains, selecting partners appropriate for changing conditions. Some fish excavate nesting burrows directly beneath coral colonies. The overhanging coral provides roof protection, while the fish maintains the burrow entrance. Males fan eggs deposited in these burrows, creating water flow that prevents fungus and delivers oxygen. The coral roots often extend into the burrow walls, creating structural reinforcement that prevents collapse. This close association benefits both species, with the fish gaining shelter and the coral receiving nutrients from fish waste, concentrated in the burrow. Certain corals produce fluorescent proteins that may protect against excessive light. These proteins absorb harmful wavelengths and re-emit them as less damaging colors, effectively converting dangerous radiation into useful light for photosynthesis. The fluorescence intensifies when corals experience light stress, suggesting an inducible defense mechanism. Some scientists believe these proteins could be manipulated to help corals survive in increasingly bright conditions. As ocean clarity changes, the proteins also create the spectacular glowing colors, visible under ultraviolet illumination, that has made some reefs famous among night divers. Some coral reefs generate their own weather patterns through evaporative cooling. Large reef platforms heat during the day and cool at night, creating temperature differences that drive local air movements. Moisture evaporating from exposed reef flats during low tides creates humid air masses that influence cloud formation. In some island systems, these reef-driven patterns contribute to predictable afternoon rain showers that nourish terrestrial ecosystems. The connection between reef health and local weather creates feedback loops where damage to corals can alter precipitation patterns on nearby land. Certain fish species time their reproduction to coral spawning events. They release eggs and sperm into the water, simultaneously with corals, overwhelming predators with superabundant food that ensures some of both coral and fish larvae survive. This synchronized reproduction requires both groups to respond to the same environmental cues, creating coordinated events involving dozens of species. The timing must be precise, as the benefits of synchronization disappear if spawning occurs even a few hours apart. Some corals develop distinct front and back sides based on consistent current direction. The upstream face receives more food and develops more feeding polyps, while the downstream side experiences calmer water and focuses on skeletal growth. This differentiation creates colonies with distinct personalities, each face specialized for different functions. The division of labor within a single organism shows sophisticated resource allocation that improves overall fitness. Colonies growing in variable currents show less differentiation, maintaining more uniform surfaces capable of feeding from any direction. Certain reef predators hunt by creating shadows that startle prey into revealing positions. Large fish hovering above the reef cast dark shapes that move across the coral, causing small fish to dart from hiding spots. The predator tracks these movements and strikes at revealed prey. This hunting strategy uses light as a tool, demonstrating understanding of how shadows influence prey behavior. The technique works best during midday when sun angles create the sharpest shadows, and predators concentrate their hunting efforts during these optimal periods. Some coral polyps vary their tentacle extension based on plankton abundance. When food is scarce, they extend tentacles maximally to increase capture surface. During plankton blooms, they partially retract tentacles since food comes so abundantly that full extension becomes unnecessary. This responsive behavior conserves energy while maintaining feeding efficiency across varying conditions. The ability to sense plankton concentration and adjust accordingly shows active monitoring of the environment and flexible behavioral responses. Certain corals create chemical gradients that attract larvae to settle near, but not directly on their tissues. The gradients guide larvae to adjacent bare substrate where new colonies can establish without competing with parents. This chemical shepherding promotes local dispersal that keeps offspring nearby while preventing overcrowding. The result is often an expanding ring of colonies around the original parent, creating distinctive spatial patterns that reflect these chemical influences. Some reef fish change their schooling behavior based on coral structure complexity. In areas with dense, complex coral, they swim in loose aggregations that allow quick access to shelter over simple, low-relief reefs. The same species form tight schools that provide protection through numbers, since shelter is scarce. The flexibility shows that schooling behavior responds to habitat structure rather than following fixed patterns. Fish assess their surroundings and adopt strategies appropriate for available cover. Certain corals host commensal crabs that steal food from polyp mouths. These tiny crabs live permanently within coral branches and snatch particles from feeding polyps. Despite this theft, the corals tolerate the crabs, perhaps because they provide other benefits such as cleaning or defense. The relationship borders between parasitism and mutualism, with the balance of costs and benefits determining whether the association helps or harms the coral. Different coral species show varying tolerance for these food thieves. Some coral colonies direct growth toward gaps in surrounding cover. When space opens due to neighbor death or disturbance, corals accelerate growth toward the vacancy. This directed expansion allows rapid colonization of newly available substrate. The ability to sense and grow toward open space requires detecting light gradients, chemical signals, or water flow patterns that indicate opportunity. Colonies that respond quickly to vacancies gain competitive advantages, capturing space before other species arrive. Certain fish perform maintenance cleaning on their home corals by removing sediment. Using their mouths and fins, they brush away particles that settle on coral surfaces, preventing smothering. The cleaning behavior appears deliberate rather than incidental to other activities. The fish return regularly to the same corals, maintaining them as carefully as they might groom themselves. This extended phenotype cleaning shows investment in habitat quality that benefits the fish indirectly through improved coral health. Some corals reduce their tissue thickness during stressful periods to conserve energy. The thinner tissue requires less maintenance and allows the coral to survive on reduced resources. Once conditions improve, tissue thickens again, returning to normal function. This reversible atrophy represents another stress response in the coral's survival toolkit. The ability to shrink and expand permits persistence through difficult periods that would kill less flexible species. Certain coral larvae can distinguish between living and dead coral substrate. They preferentially settle on living coral skeleton, recently exposed by partial colony death, rather than on long-dead rubble. The chemical signatures of fresh skeleton apparently signal suitable habitat, where the species previously succeeded. This selectivity improves settlement success, since recently dead areas likely still provide favorable conditions. Larvae settling on long-dead substrate face more uncertainty about current suitability. Some reef sponges and corals compete by overgrowing each other in slow-motion battles. Sponges creep across coral surfaces, smothering polyps as they advance. Some corals respond by deploying digestive filaments that kill sponge tissue. The battles can last months or years, with victory determining which organism controls valuable substrate. The outcomes depend on species identities, environmental conditions, and sometimes chance events that tip the balance one direction or the other. Certain corals produce more mucus during spawning periods to prevent self-fertilization. The mucus helps wash sperm away from the releasing colony, ensuring that eggs encounter sperm from other colonies rather than their own. This enforced outcrossing maintains genetic diversity and reduces inbreeding depression. The temporary increase in mucus production requires energy investment, but provides long-term benefits through healthier offspring. Some fish cultivate specific algae types by grazing selectively on unwanted species. Through repeated visits and careful cropping, they maintain patches dominated by preferred algae that provide better nutrition or easier processing. The cultivation requires memory of garden locations, ability to distinguish between algae types, and consistent effort to suppress competitors. The resulting gardens provide reliable food sources that give farming fish advantages over those that feed randomly. Certain corals accelerate their calcification rates during optimal feeding periods. When plankton abundance allows high feeding rates, the extra energy goes directly into skeleton building. During lean periods, calcification slows even if other conditions remain favorable. This tight coupling between feeding and skeleton production shows that coral growth depends heavily on food availability, not just light, for their algae partners. The relationship between nutrition and calcification affects how corals respond to environmental changes that alter plankton availability. Some coral polyps remain inactive for days during unfavorable conditions. Rather than struggling against stress, they enter quiescent states that minimize energy expenditure. Tentacles stay retracted, metabolic rates drop, and the polyp essentially waits for improvements. This patience allows survival through temporary difficulties without wasting resources on futile activity. When conditions improve, the polyps resume normal function within hours, having weathered the stress period efficiently. Certain reef fish recognize individual coral colonies and return to the same ones nightly. Memory of specific coral structures guides them through darkness to reliable shelter. The recognition might be based on visual landmarks, spatial memory, or even distinctive chemical signatures of individual colonies. This site fidelity creates strong associations between specific fish and particular corals, with the same pairs reuniting night after night for months or years. Some corals modify their skeletal architecture in response to sedimentation rates. In areas with high sediment deposition, they build smoother surfaces that shed particles more easily. Where sediment is minimal, they produce more complex structures with deeper crevices. This architectural plasticity optimizes the coral's form for local conditions, reducing the energy needed to keep surfaces clean, while maximizing structural benefits of complex growth forms where sediment allows. Certain corals maintain different algae populations in different parts of their colonies. Sunday-exposed tops host heat-tolerant strains, while shaded sides house algae adapted to low light. This within-colony diversity provides insurance against disturbances and optimizes energy production across varying light conditions. The coral actively manages these different populations, creating a patchwork of symbiotic partnerships within a single organism. Some reef octopuses change not just color, but skin texture to match coral surfaces. They develop bumps, ridges, and rough patches that replicate the three-dimensional structure of coral, creating camouflage that works from any angle. The transformation happens in seconds, controlled by muscles that alter skin topology. This ability makes them nearly impossible to detect when motionless against coral backgrounds, providing both hunting advantages and predator protection. Certain corals release chemical signals when beginning to spawn that trigger neighbors to release gametes simultaneously. The chemical coordination ensures that eggs and sperm from different colonies meet in the water, maximizing fertilization success. The signals spread across reefs in waves, synchronizing spawning across large areas. This chemical communication creates the mass spawning events that characterize many reef systems, turning individual reproduction into coordinated community phenomena. Some fish learn the daily schedules of coral polyp extension and time their feeding accordingly. They arrive at specific corals when polyps extend fully, providing maximum hunting opportunities. The learning requires memory of both locations and timing, with fish maintaining mental schedules for multiple coral colonies across their territories. This temporal knowledge allows efficient foraging that targets prey when most abundant and accessible. Certain corals can reverse partial bleaching by acquiring new algae symbionts from the water. After losing their original partners, they capture free-living algae types that may be more stress-tolerant. This flexibility in partner choice provides a mechanism for adaptation to changing conditions. The replacement process takes weeks to months, during which the coral survives on reserves and minimal photosynthesis. Success depends on encountering suitable algae and environmental conditions that allow the new partnership to stabilize. Some coral colonies develop distinct personalities in their responsiveness to disturbance. Individual colonies of the same species show consistent differences in how quickly they retract from threats, how long they remain contracted, and how vigorously they respond. These personality-like traits may reflect genetic differences, past experiences, or varying health states. The variation means that even genetically identical clones can behave differently based on their individual histories. Certain fish actively farm detritus, accumulations on coral surfaces. They defend patches where organic particles collect and periodically harvest the bacteria and microorganisms growing on the detritus. The farming behavior includes territoriality, regular maintenance, and selective harvesting that maintains the detritus garden in optimal condition. This sophisticated resource management shows planning and understanding of ecological processes that support sustained food production. Some corals survive fragmentation better when broken during certain seasons. Fragments generated during warm growing seasons establish more successfully than those created during cooler periods. The seasonal variation in fragment survival affects how storms influence population structure, with summer storms potentially increasing coral cover, while winter storms cause net losses. Understanding these patterns helps explain seasonal dynamics in coral communities and how disturbance timing influences long-term outcomes. Certain coral polyps share food resources across the colony through internal connections. A polyp that captures a large meal distributes nutrients to neighbors through tissue networks, evening out the benefits of sporadic feeding success. This communal sharing improves colony survival and growth by ensuring that all polyps receive some nutrition, even if feeding success varies spatially. The translocation of nutrients shows cooperation among polyps that benefits colony-level fitness. Some reef predators create feeding opportunities by stirring sediments near coral bases. The disturbed sediments flush out small invertebrates hiding in the substrate, making them vulnerable to predation. This technique demonstrates tool use, with the predator manipulating the environment to reveal prey. Other fish learn to follow these sediment stirrers, taking advantage of the exposed prey to feed without expending effort on their own excavations. Certain corals extend specialized tentacles at night that differ from day feeding structures. The night tentacles may be longer, more sensitive, or armed with different toxins suited to nocturnal plankton. The daily transformation between tentacle types shows sophisticated temporal niche partitioning, with the coral optimizing its capture structures for different prey communities that appear at different times. This flexibility expands the coral's dietary breadth and improves overall feeding efficiency. Some fish deposit eggs in coral crevices, then seal the entrance with sand and debris. The barricade protects developing eggs from predators while allowing water circulation. Parents maintain the seal until eggs hatch, then excavate the entrance to allow larvae to emerge. This engineering behavior shows planning, tool use, and extended parental care unusual among reef fish. The specific coral structures chosen for these nests possess particular entrance sizes and crevice depths that suit the behavior. Certain corals detect shadows of approaching predators and preemptively contract. The rapid response to sudden darkness suggests visual or light-sensing capabilities, more sophisticated than simple photosensitivity. The polyps distinguish between harmless shadows of passing fish and the looming shapes of actual predators, responding only to genuine threats. This discrimination ability reduces unnecessary contractions that would interrupt feeding without providing safety benefits. Some coral larvae delay metamorphosis if they cannot find suitable substrate. Rather than settling on poor quality surfaces, they extend their swimming period, continuing to search even as energy reserves deplete. This bet on finding better habitat makes sense when poor settlement sites guarantee death anyway. The extended larval period allows some individuals to reach distant reefs, promoting long-distance dispersal that connects separated populations. Certain fish learn which corals host the most productive gardens of film algae. They preferentially graze these high-quality locations, visiting them repeatedly while ignoring less productive corals. The selective feeding shows assessment of food quality and memory of rewarding locations. Over time, this preferential grazing can influence the distribution of algae types across reefs, creating patterns that reflect fish feeding choices rather than random algae settlement. Some corals alternate between sexual and asexual reproduction based on conditions. When the environment favors growth and expansion, they fragment and spread clonally. During stable periods, they invest in sexual reproduction that generates genetic diversity. This flexible reproductive strategy allows rapid colonization when space is available, while maintaining evolutionary adaptability through genetic recombination. The ability to switch modes shows sophisticated assessment of when each strategy provides greater benefits. Certain coral colonies coordinate defensive responses across their entire surface. When one section suffers attack, chemical signals spread through the colony, triggering defensive measures in areas not yet threatened. This colonial nervous system allows rapid, unified responses to local threats. The coordination improves defense efficiency and shows that coral colonies function as integrated organisms, despite being composed of many individuals. Some reef fish remove parasites from their own bodies by rubbing against specific coral types. The rough coral surfaces and possibly chemical compounds in coral mucus help dislodge unwanted organisms. The fish return repeatedly to the same coral heads for these grooming sessions, suggesting learned associations between particular corals and effective parasite removal. The behavior creates wear patterns on heavily used corals, marking them as cleaning stations recognized by multiple fish individuals. Certain corals grow more rapidly in turbulent water, despite the increased risk of breakage. The enhanced flow delivers more food and nutrients, providing energy that outweighs the costs of occasional damage. These species invest in flexible structures that bend, rather than break, allowing them to exploit high-energy environments that other corals avoid. The strategy works where fragments that do break off can establish new colonies, turning losses into reproductive opportunities. Some coral polyps respond to touch by neighboring polyps differently than to random contact. They recognize coordinated movements of connected neighbors and ignore them, while responding defensively to touches from separate organisms. This discrimination ability allows normal colonial function while maintaining vigilance against actual threats. The recognition system operates through chemical or mechanical signals that distinguish self from non-self at the polyp level. Certain fish time their migrations to avoid periods when coral spawning clouds the water. The reduced visibility during spawning makes hunting difficult and increases predator vulnerability. By leaving reef areas before spawning occurs, these fish avoid the disadvantages of murky water while returning after conditions clear. The timing requires accurate prediction of spawning dates based on environmental cues, showing sophisticated temporal awareness. Some corals recover from bleaching faster when surrounded by healthy neighbors. Chemical signals or shading from intact corals may reduce stress on recovering individuals. The neighborhood effect creates spatial patterns where bleaching damage spreads or contracts based on the health of surrounding colonies. Reefs with heterogeneous bleaching responses show better overall recovery than those where all corals bleach simultaneously. highlighting the importance of diversity in community resilience. Certain coral larvae preferentially settle on crustose coralline algae. The algae provide chemical cues that signal suitable substrate and may offer benefits, such as protection from grazing or improved surface chemistry for attachment. Reefs lacking these algae show reduced coral recruitment, demonstrating the importance of these foundation species for maintaining coral populations. The partnership between coral larvae and coralline algae shows how reef communities depend on interactions between very different organisms. Some fish excavate the substrate beneath corals to create expanded shelter spaces. The excavations undercut coral bases, creating caves that provide more protection than the coral structure alone would offer. The engineering activity risks destabilizing the coral but creates superior habitat for the excavating fish. The behavior represents a trade-off between habitat quality and the risk of destroying the protective coral through undermining. Certain corals show seasonal patterns of tissue thickness independent of temperature. Thickest tissues develop during periods of maximum plankton availability, regardless of when that occurs in different reef systems. The pattern shows that corals track food abundance and allocate resources to growth when feeding is optimal. This connection between tissue thickness and feeding success provides a visual indicator of nutritional status that researchers can assess without destructive sampling. Some coral colonies maintain living tissue on portions of the skeleton that receive no direct sunlight. The shaded tissues survive through nutrients translocated from sun-exposed areas, showing the communal resource sharing that allows colonies to maintain their full structure. The shaded polyps may perform other functions such as reproduction or defense that benefit the colony even though they cannot photosynthesize efficiently. This division of labor creates colonies with specialized regions adapted for different tasks. Certain fish learn the territorial boundaries of coral-dwelling damselfish and navigate around them. Rather than confronting aggressive defenders, they memorize invisible borders and swim paths that avoid conflict. This cognitive mapping of social boundaries shows sophisticated spatial memory that includes not just physical structures but behavioral zones. The mental maps get updated as damselfish shift territories or new individuals establish claims, requiring constant learning and adjustment. Some corals produce excess zooxanthellae that leak from their tissues into the water. These released algae may be taken up by neighboring corals or by larvae searching for symbiotic partners. The leakage creates a shared pool of symbionts within reef communities, potentially allowing rapid partner switching when conditions change. This algae sharing network connects coral colonies through the exchange of their most important partners, creating dependencies and opportunities across the reef. Certain coral polyps feed more aggressively after detecting chemical traces of injured prey. The scent of damaged organisms triggers increased tentacle extension and more vigorous capture responses. This reactive feeding allows corals to take advantage of moments when prey is abundant or vulnerable. The chemical sensitivity shows that corals actively monitor their surroundings for feeding opportunities, rather than capturing food passively. Some reef fish follow cleaning shrimp to coral heads where parasites accumulate. The shrimp's presence signals productive hunting grounds, where the fish can feed on parasites disturbed during cleaning activities. This commensal relationship requires the fish to track shrimp movements and understand the association between cleaners and concentrated food. The following behavior demonstrates learning and prediction that improves foraging efficiency. Certain corals develop stronger skeletons in areas with frequent storm damage. The reinforced structures resist breakage better than colonies from calm environments, showing adaptive responses to local wave climates. The strengthening involves both denser mineral deposition and modified architecture that distributes stress more evenly. Colonies transplanted from rough to calm waters gradually reduce skeleton density over subsequent years, demonstrating the reversible nature of this adaptation. Some coral larvae can detect and avoid water that recently experienced bleaching events. Chemical signals from stressed reefs apparently warn larvae that conditions are unfavorable for settlement. This avoidance behavior helps larvae bypass degraded areas in favor of healthier reefs where survival prospects are better. The ability to assess reef health from a distance provides critical information that influences settlement decisions with lifetime consequences. Certain fish defend coral heads primarily during spawning seasons, when eggs need protection. Outside reproductive periods, the same fish show little territorial behavior and wander freely. The seasonal shift in behavior shows flexible responses to changing needs, with territoriality emerging only when the benefits of defended space outweigh the costs. The coral benefits from year-round protection in some systems, but only seasonal defense in others. Some corals reproduce asexually by producing tiny clonal polyps that detach and drift to new locations. These polyp balls, called polyp bailout, represent a stress response that allows the coral to escape unfavorable conditions. Each drifting polyp can potentially establish a new colony if it settles in a better environment. The behavior represents a bet that conditions elsewhere might be superior to current circumstances, trading certain death for a chance at survival. Certain coral colonies show preferences for growing toward dominant current directions, even when other orientations receive equal light. The preference reflects the importance of food delivery through water flow, which may outweigh light optimization for corals with abundant algae partners. Colonies oriented correctly for flow often outgrow misaligned neighbors, demonstrating the fitness benefits of proper positioning. Some reef predators hunt by waiting beneath overhanging corals for prey to swim past. The ambush strategy uses coral architecture to conceal the predator's position while providing clear views of surrounding water. Prey swimming in open areas above the reef fail to notice motionless hunters in shadows below. The technique requires patience and precise timing, with attacks launched only when prey moves within strike range. Certain corals increase their investment in chemical defenses when neighboring corals are attacked. The enhanced defenses prepare the colony for possible assault by the same predators or diseases affecting neighbors. This preemptive response shows that corals integrate information about threats in their vicinity and adjust their resource allocation accordingly. The ability to learn from neighbors' experiences improves survival, even though it requires diverting energy from growth to defense. Some fish perform cleaning services, ignoring others entirely. The selectivity may reflect learned associations between certain corals and profitable parasite loads, or preferences for corals whose structure facilitates cleaning activities. The cleaners return repeatedly to preferred coral types, creating predictable cleaning stations that client fish learn to visit. This specialization creates dependencies where particular coral species become important for maintaining fish health. Certain coral larvae settle gregariously, with presence of recently settled individuals attracting additional larvae. The aggregative settlement creates clusters of young corals that may later compete intensely as they grow. The behavior makes sense if larval presence indicates suitable conditions, though it also increases local competition. The balance between benefits of settling in proven habitat and costs of crowding determines whether gregarious settlement improves fitness. Some corals extend feeding tentacles only on portions of the colony exposed to current flow. Shaded or downstream sections remain contracted, conserving energy since food delivery to these areas is minimal. This selective extension shows that colonies don't function as simple units, but as collections of polyps responding individually to local conditions. The pattern creates colonies with active and dormant zones that shift as currents change direction. Certain fish remove and eat the tube feet of sea stars attempting to feed on corals. This defensive behavior protects the coral while providing nutrition to the fish. The relationship benefits both partners, with the coral gaining protection and the fish receiving food and a safe territory. The specificity of the partnership, with particular fish species defending specific coral types from particular predators, shows coevolution that has refined the mutualism over time. Some coral colonies surviving bleaching events subsequently show reduced growth rates for years. The long-term impacts of stress persist even after the colony recovers visually, affecting reproductive output and competitive ability. This legacy effect means that bleaching impacts extend far beyond the immediate event, potentially weakening coral populations through reduced reproduction and slower space capture. Understanding these delayed effects is crucial for predicting long-term community changes. Certain corals coordinate their spawning times with specific moon phases that optimize larval survival. The timing ensures that larvae spend their vulnerable early days during neap tides when currents are weakest, reducing the risk of being swept into unsuitable habitat. The precision of this timing, maintained across entire populations, shows that natural selection has fine-tuned reproductive schedules to match oceanographic patterns. Even slight shifts in spawning time can dramatically affect offspring survival. Some fish use coral branches as reference points for elaborate courtship displays. Males perform dances around specific coral structures, using them as stages that make their movements more visible to females. The coral choice may reflect preferences for particular heights, branch densities, or locations within the territory. Females assess both the quality of the dance and the coral structure itself, which indicates male territory quality. Certain coral polyps alter their feeding behavior based on time since last successful capture. Recently fed polyps become less responsive to passing prey, while hungry polyps react more aggressively. This graduated response conserves energy by preventing wasteful strikes when the polyp has adequate nutrition. The system requires memory of feeding state at the individual polyp level, showing surprising cognitive capacity in these simple organisms. Some corals develop different morphologies when growing in waves versus steady currents. Wave-swept individuals produce more flexible structures that bend with surge, while current-adapted colonies build more rigid forms oriented into the flow. The architectural differences reflect optimization for different hydrodynamic environments. Genetic studies show these variants are often the same species expressing different phenotypes rather than distinct genetic lineages. Certain fish learn to associate specific coral species with high-quality shelter. They preferentially settle near these corals and defend territories centered on them. The learned preferences are culturally transmitted, with young fish learning which corals provide best shelter by observing the choices of older individuals. This cultural knowledge affects community structure by creating consistent associations between fish and coral species across generations. Some coral colonies retain genetic memory of stress events through epigenetic changes. Colonies experiencing severe stress modify how their genes are expressed without changing the DNA sequence itself. These modifications can persist for years and may even transfer to offspring, potentially allowing corals to pass stress tolerance to the next generation. The mechanisms are still being discovered, but suggest corals possess adaptive capacities beyond traditional genetic evolution. Certain corals produce distinct chemical signatures that fish learn to recognize. Individual colonies may have characteristic scents based on their specific microbial communities and metabolic outputs. Fish use these signatures to navigate the reef, relocating favorite corals, even in darkness or murky water. The chemical recognition requires olfactory memory and discrimination abilities that allow distinguishing between very similar scent profiles. Some reef fish modify their foraging patterns based on coral health status. They avoid feeding near stressed corals, possibly recognizing chemical signals of distress or associating unhealthy corals with reduced prey availability. This behavioral response creates spatial patterns where degraded reef areas receive less fish activity, potentially affecting nutrient cycling and ecological processes that depend on fish presence. Certain coral larvae can delay settlement for weeks while drifting in search of ideal conditions. Extended larval durations allow dispersal across vast distances, potentially connecting reef systems hundreds of miles apart. The larvae subsist on stored energy reserves, with some species possessing enough reserves to drift for over a month. This long-distance dispersal capability helps maintain genetic connectivity between distant reefs and allows recolonization of disturbed areas. Some corals fragment intentionally during reproduction, with specific zones of weakness that break preferentially. The controlled fragmentation produces pieces of optimal size for dispersal and establishment. Unlike random breakage from storms, this purposeful division occurs during seasons when fragment survival is highest. The strategy combines vegetative reproduction with dispersal benefits, usually associated with larvae. Certain fish territories center on clusters of multiple coral species rather than single colonies. The diversity within territories may provide varied shelter options, multiple food sources, or reduced risk from coral-specific diseases. Territory holders defend the entire cluster against intruders, effectively protecting a miniature reef ecosystem. The complexity of these territories suggests that fish value habitat diversity as well as sheer quantity. Some coral polyps share captured food preferentially with neighbors showing signs of starvation. The directional nutrient flow towards struggling polyps shows a form of colony-level resource management that prioritizes keeping all members alive. This cooperative feeding improves colony survival. During periods when only some polyps successfully capture prey, the system requires communication of nutritional status across the colony network. Certain corals modify their pigmentation based on neighboring corals' colors. Colonies growing beside brightly colored neighbors often develop enhanced pigments themselves, possibly to compete for attention from beneficial fish or to optimize light environments. The responsive pigmentation shows that corals monitor and react to the visual environment created by their neighbors. The phenomenon creates color patterns at the community level that emerge from individual colony responses. Some fish learn complex routes through coral mazes that minimize swimming distance. The mental maps include three-dimensional information about passages through coral structures, allowing efficient navigation through intricate habitats. Fish traversing these routes swim with confidence, making turns before landmarks become visible, indicating memory-based navigation rather than reactive following of visual cues. The spatial cognition required rivals that demonstrated by vertebrates with much larger brains. Certain coral colonies respond to repeated non-damaging disturbances with reduced sensitivity. This habituation prevents energy waste on defensive responses to harmless stimuli. The learning occurs over days to weeks and can be specific to particular disturbance types, showing discrimination and memory at the colony level. The adaptive value lies in allowing corals to ignore harmless but frequent disturbances, like brushing fish, while maintaining responsiveness to genuine threats. Some corals grow photosynthetic symbionts in specialized structures that maximize light capture. These symbiont chambers position algae optimally while protecting them from excessive radiation. The architectural sophistication shows evolutionary refinement of the coral-algae partnership through structural adaptations that benefit both partners. Different coral species have evolved different chamber designs, each optimized for their particular light environments. Certain fish perform distraction displays to lead predators away from coral-sheltered eggs. The parent fish feigns injury or vulnerability, drawing predator attention while swimming away from nesting sites. Once the threat moves sufficiently far from the eggs, the fish escapes to safety. This deceptive behavior shows planning and understanding of predator psychology, with the fish deliberately risking itself to protect offspring it will never see again after hatching. Some coral colonies develop chimeric forms by fusing with neighboring colonies of the same species. The fusion creates individuals with multiple genetic identities, sharing a single skeleton. These chimeras may possess advantages through combining genetic diversity within one colony, potentially improving disease resistance or stress tolerance. The acceptance or rejection of fusion partners appears to involve recognition systems that assess genetic relatedness. Certain corals increase skeleton porosity during rapid growth phases to reduce construction costs. The lighter structure allows faster expansion, but provides less strength. Once growth slows, the coral reinforces the porous skeleton with additional mineral deposits. This two-phase construction strategy balances the competing demands of rapid space capture and long-term structural integrity. The approach shows sophisticated resource allocation across different life stages. Some fish cultivate coral growth by removing algae competitors from preferred coral species. The grooming behavior benefits both partners, with corals gaining relief from competition and fish maintaining high-quality shelter. The selectivity of the grooming, with fish targeting specific algae types while ignoring others, shows fine-scale manipulation of the coral's environment. This gardening behavior creates conditions that favor coral growth without the fish receiving immediate food rewards. Certain coral larvae assess substrate quality by testing attachment strength before committing to metamorphosis. They perform temporary attachments and detach if the bond is weak, continuing their search for more suitable surfaces. This quality control improves settlement success by ensuring larvae only commit to substrates where secure attachment is possible. The discrimination requires mechanical sensing and decision-making that prevents wasted investment in poor locations. Some corals develop localized immune responses that protect injured areas without activating colony-wide defenses. This targeted response conserves energy by limiting defensive measures to zones actually under threat. The spatial precision of the immune system shows sophisticated internal communication that differentiates between local and systemic threats. The efficiency gained through localized responses allows corals to maintain growth and reproduction while managing injuries. Certain fish modify their swimming styles when approaching coral with extended polyps. They move more slowly and avoid sudden movements that might startle the coral into retraction. The considerate approach maintains access to shelter while preventing wasteful defensive responses from the coral. The learned behavior shows that fish understand the connection between their movements and coral reactions, adjusting behavior to maintain beneficial conditions. Some coral colonies create microenvironments within their branches that differ significantly from surrounding water. Temperature, flow, and chemistry within dense branches can vary from ambient conditions, creating refuges where unusual organisms survive. The microenvironments arise from the colony's physical structure, modifying local conditions through shading, flow baffling, and metabolic outputs. These hidden habitats contribute to overall reef diversity by supporting species that couldn't survive in typical reef environments. Certain corals release antimicrobial compounds into their mucus that suppress specific pathogens. The chemical defenses are inducible, increasing when the coral detects pathogen presence. This targeted response allows corals to fight infections without maintaining costly defenses constantly. The specificity of antimicrobial production suggests recognition systems that identify different pathogen types and deploy appropriate chemical weapons. Some fish time their feeding bouts to match coral polyp extension patterns. They learn when specific corals extend tentacles and hunt the small organisms attracted to the feeding polyps. The timing requires memory of multiple coral schedules and the ability to plan movements to arrive at each coral when feeding opportunities are optimal. The complex scheduling demonstrates impressive cognitive abilities applied to maximizing foraging efficiency. Certain coral colonies respond to water flow by orienting their polyps into the current. The directional sensitivity allows them to position feeding tentacles optimally for prey capture. Individual polyps can rotate slightly within the skeleton to achieve better orientations, showing active positioning rather than passive acceptance of random arrangements. The collective orientation of thousands of polyps creates colonies with coordinated feeding surfaces all facing into productive currents. Some corals survive extreme low tides by producing thick mucus layers that retain moisture. The mucus coating prevents desiccation during aerial exposure, allowing survival in intertidal zones. The protective layer also reflects sunlight, reducing heat stress during exposure. Once the tide returns, the mucus sloughs off, carrying away any organisms or debris that accumulated during the exposure period. This dual function shows how a single adaptation can address multiple stresses. Certain fish learn to predict coral spawning and gather to feed on the released gametes. The predictive ability requires memory of previous spawning events and recognition of environmental cues that precede spawning. Fish that successfully predict spawning gain access to abundant food resources unavailable to those that fail to anticipate the events. This cognitive forecasting provides significant fitness advantages during brief periods of resource abundance. Some coral colonies develop distinct growth sectors with different skeletal properties. The sectors may differ in density, porosity, or trace element composition, creating internal heterogeneity within single colonies. The variation may reflect seasonal changes in growth conditions or responses to localized disturbances. The sectored growth creates internal complexity that records the colony's environmental history in spatial patterns, rather than simple chronological layers. Certain corals maintain populations of diverse bacteria that produce antibiotics continuously. The microbial shield provides ongoing protection against pathogenic organisms attempting to colonize coral surfaces. The diversity of antibiotic-producing bacteria ensures broad-spectrum defense against multiple pathogen types. The coral cultivates this protective community by providing nutrients and habitat, creating a farmed immune system that operates outside the coral's own tissues. Some fish defend multiple coral colonies simultaneously, dividing time between separated territories. The multi-site defense requires memory of locations, assessment of which sites face greatest threat, and allocation of effort across territories. The strategy makes sense when no single coral provides all necessary resources, forcing fish to maintain access to multiple locations. The territorial complexity demonstrates spatial memory and time management that optimizes access to scattered resources. Certain coral larvae synchronize their settlement, timing with optimal tidal conditions. By settling during specific tidal phases, they improve their chances of remaining on suitable substrate, rather than being dislodged by subsequent currents. The timing precision requires larvae to track tidal cycles during their drifting period and delay or accelerate metamorphosis to match favorable moments. This temporal control over settlement shows sophisticated environmental tracking by tiny, simple organisms. Some corals develop enhanced structural support, specifically beneath heavy branches. The reinforcement occurs exactly where needed to prevent collapse, showing remarkable precision in resource allocation. The targeted strengthening suggests that corals sense structural stress and direct skeleton construction to address weak points. This responsive architecture allows building large, complex forms without wasting resources on uniform overstrengthening. Certain fish learn to associate particular coral morphologies with specific shelter qualities. Branching corals provide different refuge from plate corals or massive forms, and fish develop preferences based on their specific needs. Young fish learn these associations through experience, with preferences strengthening as they survive predator encounters in different shelter types. The learned preferences create patterns where fish species segregate among coral types based on matching shelter requirements to coral architecture. Some coral colonies reduce metabolic rates during extended stress periods. The deliberate slowdown conserves energy and reduces production of damaging metabolic byproducts. The strategy accepts slow starvation as preferable to rapid death from metabolic damage. Colonies that successfully manage this metabolic depression can survive stress periods that kill those maintaining higher activity levels. The recovery after stress depends on the colony retaining enough reserves to resume normal function. Certain corals respond to injury by accelerating growth in undamaged areas. This compensatory growth helps offset losses from damaged zones, maintaining overall colony size and function. The response requires communication of injury status across the colony and reallocation of resources toward healthy zones. The strategy works when injuries are localized, allowing the colony to recover total size, even while damaged areas heal slowly. Some fish learn the three-dimensional structure of coral colonies in complete darkness. They navigate through complex coral branches at night, using lateral line sensing and memory of daytime visual surveys. The mental maps persist accurately enough to allow rapid movements through intricate passages without light. This multimodal mapping shows sophisticated spatial cognition that integrates information from different sensory systems into unified representations. Certain coral larvae can survive extended periods without food by recycling internal resources. The metabolic efficiency allows dispersal across great distances, even when plankton food is scarce. The larvae essentially digest their own tissues in controlled ways that maintain critical functions while sacrificing less essential structures. This self-cannibalization buys time to find suitable settlement habitat at the cost of reduced post-settlement vigor. Some corals produce chemical compounds that promote their own recruitment by attracting conspecific larvae. The self-recruitment enhances local population density and creates clusters of related individuals. The chemical attraction works over short distances, guiding larvae that have already reached the general area to optimal settlement sites. This chemical shepherding improves recruitment success compared to random settlement across available substrate. Certain fish learn individual coral colonies' defensive responses and adjust approaches accordingly. Corals that retract readily are approached cautiously, while those that remain extended receive less careful treatment. The learned association between individual corals and their behaviors shows impressive memory capacity when fish interact with potentially hundreds of different colonies. The knowledge allows efficient movement through the reef without triggering unnecessary defensive responses. Some coral colonies coordinate their symbiont populations across connected individuals. When colonies fuse, their algae populations mix and homogenize over time, creating consistent symbiont communities across the chimeric structure. The mixing may provide benefits through averaging stress responses or combining different algae strains. The phenomenon shows that symbiont management occurs at scales beyond individual polyps, involving colony-wide or even population-level processes. Certain corals increase water flow through their tissues by coordinating ciliary beating patterns. The generated currents improve gas exchange and nutrient delivery beyond what ambient water movement provides. The coordination requires some form of communication between cells to maintain synchronized beating. The enhanced internal circulation allows corals to maintain high metabolic rates, even in calm water where external flow is minimal. Some fish develop traditional knowledge of coral quality passed between generations. Young fish learn which coral species provide best shelter not through individual experience, but by observing and copying the preferences of older individuals. This cultural transmission creates stable associations between fish and coral types that persist even as individual fish are replaced. The traditions may take many generations to change, creating community stability that depends on social learning. Certain coral larvae respond to specific light wavelengths that indicate optimal settlement depth. Blue light signals deep water, while red light indicates shallows, allowing larvae to navigate vertical gradients even without direct depth sensing. The wavelength preference guides larvae toward depths where their species succeeds, improving settlement success. The mechanism provides orientation in open water, where other depth cues are unavailable. Some corals develop defensive structures only after experiencing predation. The induced defenses emerge over weeks following attacks, showing a form of learning at the colony level. The defensive investment occurs only when needed, conserving resources when predation is absent. The response shows that corals monitor their environment for threats and adjust their phenotypes accordingly, displaying plasticity that optimizes the trade-off between growth and defense. Certain fish perform maintenance behaviors on corals during specific times of day. Morning grooming removes sediment accumulated overnight, while evening sessions might focus on removing algae that grew during the day. The scheduled maintenance shows time-based task differentiation that keeps corals clean through complementary morning and evening. The consistent timing suggests circadian control of cleaning behavior. Some coral colonies respond to climate oscillations by adjusting their growth strategies years in advance. El Niño-sensitive corals show changes in growth rates and skeletal density before warming occurs, possibly responding to subtle oceanographic precursors. This anticipatory behavior suggests that corals integrate long-term environmental trends into their growth planning, optimizing strategies for predicted rather than current conditions. The forecasting ability provides advantages when corals correctly anticipate coming changes. Now transitioning to the conclusion section. As we drift toward the close of this gentle journey, let your awareness of these remarkable reefs settle softly like sediment, finding rest on the seafloor. You have explored worlds within worlds, discovered partnerships between the smallest and largest of creatures, witnessed architecture built molecule by molecule across centuries. The coral reefs continue their patient work beneath the waves, building and sheltering and adapting in rhythms older than language. Perhaps tonight you found a favorite fact that will stay with you into dreams. Maybe it is the image of glowing corals beneath moonlight, or the thought of tiny polyps sharing food across their colony, or fish that remember every coral in their territory. Each of these small wonders is true, happening right now somewhere in the warm seas of our planet. The reefs don't rest, but their work is never hurried. They teach patience, cooperation, and the quiet power of small actions multiplied across time. If these peaceful explorations have brought you comfort, I gently invite you to subscribe so you can return whenever you need rest. A simple like helps others find their way to this quiet corner of wonder. And if you wish, share a thought in the comments about which coral fact surprised you most or what you'd like to explore in future journeys. Your presence here matters, and every small gesture of support allows these gentle voyages to continue, reaching other souls who seek learning wrapped in stillness. But there is no urgency to any of this. The channel will be here tomorrow and the day after, waiting whenever you need a soft place to land. For now, all that matters is your comfort. The reefs have shared their secrets. The polyps are extending their tentacles into the quiet current. The fish are settling into their nighttime shelters among the branches. And you, too, can rest now. Let your breathing slow to the rhythm of gentle waves. Feel your body growing heavy, as if seawater were flowing through your limbs, warm and calm. Your thoughts can drift like larvae on a patient current, moving without effort, requiring nothing of you. The colors of the reef begin to fade into soft blues and purples as depth and darkness embrace them. Every creature finds its resting place. Every polyp draws inward. The reef itself seems to exhale, releasing the day. You don't need to remember everything you've heard tonight. The knowledge will stay somewhere in the quiet chambers of your mind, ready to surface when needed. For now, just allow it all to blur and soften. Science and story, fact and feeling, all flowing together like water and light. There is nothing you need to do, nowhere you need to be, no problem that requires solving in this moment. The ocean continues its eternal conversation with the reef. Currents bring nutrient, waves bring rhythm. The moon pulls tides across the coral gardens, and everything that lives there moves to patterns established long before we learned to speak of them. You are part of this ancient system, now connected through attention and wonder to the smallest polyp, building its tiny home in the warm sea. As your consciousness grows dim, imagine yourself floating just above a coral garden at dusk. The water is perfectly warm. You can breathe without effort. Below you, the corals are settling into their night forms, some extending new tentacles, others contracting into sleep. Small fish dart into crevices. A octopus flows like liquid shadow across the reef surface. Everything is exactly as it should be. You are safe here in this imagined water. No current can carry you away. No predator threatens. You simply exist, suspended in gentle blue, watching the light fade from amber to indigo to the soft black of tropical night. And in this darkness, tiny bioluminescent sparks begin to appear. Pinpricks of light that pulse and fade, like stars in the upside-down sky. The rhythm is hypnotic, soothing, perfect. Your eyelids grow heavier with each pulse of light. Your muscles release tension with each gentle wave that passes overhead. The reef below becomes harder to see, fading into darkness. But you know it is there. You trust its presence. Millions of polyps breathing in the dark, countless small lives continuing their work, and you floating above it all, finally ready to release the day. The next video will be here when you wake, ready to carry you to another place of peaceful learning. But for now, there is only this moment, this breath, this softening. The reef whispers goodnight in chemical and current, in the settling of sand and the closing of anemone tentacles. All of nature is settling with you, finding rest after the long work of living. Let go now. There is nothing left to hold. The gentle science of sleep is calling you down to dreams where coral gardens grow inside your mind, where you swim without fear through forests of impossible color, where every creature knows your name and welcomes you home. The water holds you. The darkness embraces you. The reef releases you to rest. Good night.